Yes. Good evening, participants. I think I'm audible to you. So let me go to the 13th session. The 13th session is about machine learning approaches of drug design. Okay. Actually, this we are seeing in 13th day uh, for certificate and internship course on artificial intelligence and machine learning approaches of drug and vaccine design in cancer. Okay, before going to this 13th chapter, can we recap the last session topics? So, what are the last session topics? Is good. So, I got answer from Chintu that B cell epitope prediction, and we have discussed about adjuvants. Good. So, mention any four methods of B cell epitope prediction. So, whatever methods we have discussed. And if I ask about even tools, you should know. But any four methods, so I can type the question. Mention me about any uh, four methods of B cell epitope prediction. What else we have seen other than the B cell epitope prediction and adjuvants? We have also seen, yeah, only one is interacting. How about the others? Yet, still from the same participant, I got one method from Zintu, Gemini surface accessibility. And any other? Yes, interact, or you will be forgetting. Okay, so you have to watch the videos if you didn't attend on last session. So what are the methods? Because if you want to, whatever you study for all the participants, I can suggest you to practice. If you're not practicing, then you acquire something you can able to understand, but it won't register in your mind. I told you it can be T cells or B cells. We have a memory cells. These memory cells will try to take a memory to combat cancer or infectious cells, isn't it? So similarly, here also we should acquire memory to not to forget the skills, whatever I'm providing in hands-on experience. But, I got a Markov model. Is it so? No. But Car Plus is okay. Car Plus, Chow Fastman. Okay. So for this, everyone can go to the IEDB website. Go to the IEDB website, go for B cell epitope prediction. Then you can find all the methods. Nearly 6 methods are there in B cell epitope prediction. You know, the first method is baby bread, isn't it? Then what we have seen, Car Plus, and then we have gone for Emini, and then so many methods. Huh? So we have seen nearly 6 methods in IEDB. And subsets of T cells, we have seen if dendritic cell interacts with a native T cell, it can generate native T helper cell, then it can generate TH1 and TH2 cell. Okay, other than TH1 and TH2, we also have cytotoxic T cells, okay, which can directly go for combating cancer, but helper 1 and helper 2 cells can secrete cytokines, interleukins. These can also activate the T cells or even APCs, so that even these cells can combat the cancer indirectly. Okay, so likewise, so we have plenty of antibodies generated from B cell if B cell interacts with the T cell. Okay, so we have almost cytokines, antibodies, types of T cells and types of Chow Fastman and types of T cells. Okay, and even subsets of B cells. Okay, so all these things can combat the cancer. Okay, so mostly we have discussed about the cellular immunity as well as humoral immunity, even in the last session. Okay, so now I think I can take you to today's session on machine learning approaches of drug design. And before going to this, I also told you after predicting the epitopes, okay, of B cell, you have to go for docking, isn't it? So whatever epitopes you predicted from any virus or cancer antigenic proteins. Okay, how we can check the efficacy of these proteins, whether this is effective against humans or not? If only if you dock the predicted epitopic peptides can be from T cell or from B cell, you have to dock with appropriate human proteins, isn't it? So if you see the B cell epitopes, whatever you predicted, these epitopes have to be docked with what human proteins? If only if you dock, then only we can able to screen the best B cell epitope. Just like that, with the scores, whatever scores you get from the different methods, can't finalize your the best epitope. Okay, I told you, no, the last slides. What are the receptors I told you to take to consider? And even one or two participants are also queried, also asked the questions on the last session. What is a receptor? The last time, even some participants have asked me, no, the expansion of a particular receptor. What is that? No one is trying. See, whatever you're watching will be lead to waste. You should capture from head to tail. Very good. The answer is TLR, that is toll-like receptors. Whatever B cell epitopes you predict, you have to talk with corresponding TLRs. For example, if you are combating a bacterial pathogen, then obviously whatever epitopes you predict from the B cell, okay, you have to talk with TLR4. TLR4 is for the bacterial pathogen. And if you're having a virus, a viral infection, viral infections, then you have to talk with TLR7 or 9. Okay, so we have a corresponding toll-like receptors. If and if you talk that with a particular protein, suddenly you can finalize it. Okay, which is the best one. Okay, this is most important, but you should take care. Okay, so how to work on the projects. Now let me take you to the new chapter called machine learning approaches of drug design. Just an introduction, like I already mentioned you, I'm guiding some 3 PhD scholars. I am guiding on anti-cancer drug discovery and development, and also anti-diabetic drug discovery and development. Okay, so these are the areas I'm focusing, and my research areas of interest in Faculty of Technology in Anna University are drug design, vaccine design, pharmacognosy, and immunotechnology. Of course, I also involved in drug development and vaccine development. Recently, I'm using nanotechnology and mitochondrial targeting for the diabetes as well as for the cancer. Now, let me take you to this machine learning. What is machine learning? So, machine learning is a field of study in artificial intelligence. You see, the big circle is artificial intelligence. A part of a study is the machine learning, isn't it? So that's what a concern with the development and study of statistical algorithms that can learn from data and generalize to unseen data and thus perform tests without explicit instructions. So if you want to run something with in automation, okay, or not giving each and every manual task, then better we can go for machine learning approaches. If you see this father of machine learning, Arthur Lee Samwell, an American pioneer in the father of machine learning, is expert in the field of computer gaming and artificial intelligence. Okay, the synonym came in this period. And if you see the machine learning algorithm, there are types of machine learning algorithm: supervised learning, unsupervised learning, semi-supervised, and reinforcement. Majorly, if anyone asks about the types of machine learning algorithm, we can classify into 3 types: supervised, unsupervised, and reinforced. So what is supervised learning? How can we classify? What are the methods you can find in supervised learning? The major methods you can find is classification, the other is the regression. If you have a sample of data in a population, if you have multiple samples, and in each and every sample you have some set of data, then how to classify these data? Okay, so that's comes under supervised learning, and how to go for linear regression, how to link all this data in a linear relationship, y equals mx plus c. If it is a linear regression, you find that kind of a equation. There is also multiple regression. Okay, so if you see the supervised learning, so basically we can expect these kind of methods like linear regression, logistical regression, random forest, gradient boosted trees, support vector machines, neural networks, decision trees, neighbors, nearest neighbor, etc. And if you see this unsupervised learning, basically you find these two learning tasks. One is clustering, the other is dimensionality reduction. You remember clustering? We generally use in microarray data. If you go get a gene expression data for a cancer, there is a need to go for a clustering of data. Then only we can able to diagnose the stage of a cancer. Okay, and even if you see this classification, if you have if a patient is suffering from liver cancer, there are types of liver cancer. Okay, hepatocellular carcinoma is one of the liver cancer. So first we have to check whether particular patient is suffering from hepatocellular carcinoma. Then you have to undergo 2 or 3 tests. Might be the third test can confirm hepatocellular carcinoma, which is the final stage of liver cancer. Okay, but if the patient is suffering from third stage, okay, that is in the form of a fibrosis, okay, then even we have to classify whether is it in the third stage or not. Okay, so the classification also comes under, but generally classification comes under major classifying the data. Okay, and regression is just we can also find in protein structure prediction. We can find this especially in support vector machines. In support vector machines, where majorly we go for protein folding. Okay, this is used for determining the energy minimization, keeping a protein stable in the software environment. So for this, you can also use this kind of regressions. Okay, now if you see this clustering, I told you for microarray data population to screen the stage of a cancer. This is also used for uh, hierarchy. If you want to uh, put the protein in the form of a taxonomical classification, we can also use the clustering. There are plenty of applications in bioinformatics. I'm telling you a few examples for clustering and dimensionality reduction. Okay, so if you see this, we can see this even in principal component analysis. In the molecular dynamics simulation, if your RMSF is showing a high deviation with respect to C alpha fluctuation, then obviously you have to go for principal component analysis. Okay, so in MD simulation, you can find this kind of analysis. Likewise, we can find k-means clustering. This k-means clustering generally used for creating the phylogenetic trees. Okay, and t-distributed stochastic neighbor embedding. Of course, similarly, similar principle, similar application, association rule, instant-based learning models, ANN, genetic algorithm. You remember the genetic algorithm we also used in Autodoc for docking purpose. If you use a Autodoc tool, we are using a genetic algorithm, especially Lamarckian genetic algorithm. Okay, so and also we have for the deep learning models, especially if you want to work for pharmacophore development. Okay, in the drug design, obviously we have to go for deep learning models, and even for QACR. Okay, quantitative structure activity relationship studies, you should one should have this knowledge, deep learning models. And if you see the reinforcement learning, we have model free and model based. Okay, these are the two classes. Actually, here if you see, this is more strategic. Okay, when compared to these, and if you see this kind of methods, basically we have the algorithms like Hue learning, temporal difference, Monte Carlo research. MD simulation is majorly uh, developed based on this method, Monte Carlo. And asynchronous actor critic agents, deep Q learning, temporal difference, data set aggregation, asynchronous actor critic agent. So actually, this is actually we build the models. We build the models in the website itself. Okay, and for these models, you can practice and see whatever you have the models. Like for example, if you want to work on QSR analysis, there are few websites where the training models are already developed with the data sets. So if you have any test data set, you can just input the test data set and you can find the value for it. If it is a QSR, you predict the activity of your unknown form. So all these things, okay, this kind of developing the models and using the model already existing in the web resource. Okay, so that is model based, and there is also model free where even without creating models, you can work on these algorithms. Sorry, applications of machine learning in drug discovery. I told you in supervised majorly you have classification and regression analysis. So in classification, NLP kernel methods, NLP Bayesian classifier, support vector machines, nearest neighbor. These are some of the classification methods. What for it is used? It is used to target disease drug associations and also target drug ability based on pharmacokinetic properties and protein structure or sequence. And we also use for tissue specific biomarkers from gene expression signatures. Okay. So if you want to identify the prognostic markers, diagnostic markers, obviously there is a need to know about the classification, okay, which comes under supervised learning. And if you see the regression analysis, majorly random forest, linear regression, decision trees, sparse linear regression. These comes under like a regression analysis. What for these algorithms are used? These are used for predicting the molecular features that for the cancer drug response, and also for targets for Huntington disease and target drug ability from multidimensional data, and also gene expression data that predict clinical trial success. So the supervised learning is used for these drug design applications. And if you see the unsupervised learning, especially the clustering, you have hidden Markov model, hierarchical clustering, K-means clustering, and artificial neural networks. If you see this, this is used for de novo molecular design. We want to design a molecule, so we need this, and we need also use for deep future selection for biomarkers, future reduction in single cell data to identify cell types, especially those who are working on single cell genomics. They should have a knowledge on the cluster, cell types and biomarkers from single cell RNA data. This is also important. Okay, so of course this clustering also gives fundamentals to work on NGS. A basic machine learning system arrangement. Okay, so if you want to work on this machine learning, generally you should remember two terminologies, two technical terms. One is training data set and the other is the test data set. The training data set is what you have to develop in the software. Okay, in the machine learning software. So how the training data can be developed? It can be labeled or unlabeled. First, you should have a machine learning algorithm like artificial neural networks, hidden Markov model, decision trees. So if or you should use one appropriate machine learning algorithm for the knowledge based and for decision making. So for what application are you using? Whether you want to develop a software or a machine learning model for predicting the protein structure, or predicting the protein folding kinetics, or whether you're using it for drug design, protein drug binding. So all these things. So what is the major application? Based on that, you first develop a training data set. That means you have to populate as much data. Minimum you have to populate 100 data. So if you populate 1,000 data, whatever unknown data you're going to check with the model will give you the precise result. The larger the data set, the more precise kind of an output you'll be getting in from the data set. Okay, so this is something related to machine learning model. What is application? Okay, develop a training data set, use a test data set to predict with the training data set. Okay, and finally, you can evaluate the model. Commonly used machine learning software and tool. So most probably we are using the Keras, the Python deep learning library, scikit-learn, of course, machine learning in Python. TensorFlow, I think many of you heard about this. It's an open source software library for machine intelligence. Cloud computing. In cloud computing, you have heard about the TensorFlow, isn't it? And R programming, you heard, isn't it? The R project for statistical computing. Vika, a data mining tool in Java. MATLAB, a multi-paradigm programming language and numerical computing environment. But may also are using Theano, Python framework for fast computation. The advanced developers are using this kind of a machine learning tool. Torch for scientific computing. Okay, PyBrain, modular machine learning library for Python. Overview of molecular docking sampling and scoring flowchart. How the machine learning is used to go to perform molecular docking in softwares. So generally, how the molecular docking is happening. So the molecular docking is generally using two different algorithms, two important algorithms. One is for searching, that is a search algorithm. The other is for the scoring function. Okay, so if you want to check if you have hundreds of drugs, I mean hundreds of inhibitors, and you have a target receptor, you want to screen what is the best from the 100 inhibitors, whatever you have. So what you have to do, you have to go for the search algorithm, isn't it? So that is, you have to check which is best, like going for virtual screening, however we mentioned, or you can go for high throughput screening. So what is that virtual screening, high throughput screening, and a lead hopping? All these are, of course, a part of a molecular docking process. But we go for docking, binding with numerous compounds and coming up with a best drug, coming up with a best inhibitor, isn't it? So finally, we evaluate. So how this happened on the search algorithm generally uses systematic search, molecular dynamic shape complementarity genetic algorithms. Generally, if you want to dock multiple ligands with a receptor in Discovery Studio, there is one commercial software called Discovery Studio from BioVia. We are going to use a software called LibDoc. LibDoc is what the software tool for a search algorithm in Discovery Studio. But there is also one more algorithm called scoring function. I told you in Discovery Studio, we have one tool called LibScore. LibScore. Okay. This is LibDoc and this is not LibScore, Ligand Score. This is Ligand Score. Okay. So in the Ligand Score tool, is generally used to calculate 8 different scores. Generally, if you perform molecular docking in Autodock, you get only one score, that is internal binding energy in terms of kilocalorie per mole. So in Autodock, whatever energy you will be getting, that should be -6 and below kilocalorie per mole. Then you can tell whatever binding happened is significant with respect to energy. But if you come across the Discovery Studio or a few more softwares, commercial softwares, we will be getting some scores. We're not getting a single score, but we get some few scores. If you see this ligand score module, you'll be getting 8 different scores in ligand from the Discovery Studio. So how these 8 different scores are calculated? So actually, this is based on different force field or molecular mechanics. There are types of energy minimization protocol, isn't it? MMFF, you have CFF, you have CHARM, CHA, RMM. There are types of scoring energy minimization methods. So based on these empirical methods or knowledge-based scoring or consensus scoring, the scoring function is used to score the binding affinity, and finally it can also evaluate the docking. Okay, and finally whatever you dock, you can also evaluate through the simulation through the RMSD by predicting the RMSD after the docking. Okay, so here if you see numerous parameters are taken in consideration, like selection of target protein, selection of ligand, search for all possible orientations, locates the rotational bonds here in the algorithm, explores conformational space of ligand by keeping protein rigid, searches for structural binding complementary between protein and ligand, searches for optimum protein ligand. So a docking software is generally developed utilizing all these parameters surrounded. Okay, so just you have search algorithm scoring function, but you have to consider all these parameters to implement this algorithm. Okay, this is how a docking software works. One such important thing is that whatever you're going to dock, okay, if you should not only calculating the free energy for the protein ligand complex, but you're also calculating the favorable binding interaction. If and if your ligand is binding with the appropriate binding sites, then you can tell that is a correct inhibitor for this disease. Okay, if it is not docking with the binding cells, if it is docking with some other binding cell, then it's not. So even the scores are developed based on this kind of uh, favorable binding interaction from existing protein ligand database. You already have some data deposited in the protein ligand database. It also checks with it, and even the score is generated based on that. Okay, and also the rescoring is possible in this method. Now, let me take you to the structure-based drug design. So, in structure-based drug design, especially if you want to predict a structure of a protein, okay, we have already discussed. We have gold standard techniques like X-ray crystallography, NMR, bioelectron microscopy, X-ray diffraction. We have plenty of methods, isn't it? So whatever methods you use, okay, generally you get if it is an X-ray, you just going to get in black and white spots. Whatever you get a black or white spots, the diffraction pattern, you pass it to the software to get an electron density maps. So whatever electron density maps it generates, if you pass it to the simulation software, so it generates a 3D structure. So we can find the solid ribbon kind of a model from the visualizer. Okay, so we generally get a black and white spots. Like however we get X-ray images. If you go for scanning, okay, especially taking X-ray. So if a patient is suffering from lung cancer, so obviously whatever the X-ray image, if you see only black and white spots and some gray spots, isn't it? The same spots even you can find in the protein crystal. If you crystallize and the 3D structure, whatever you get, you from the diffraction pattern only, you'll be getting this computational PDB structures. And if you see this, the kind of target binding, if your ligand is going to bind in that particular target, then obviously we can able to check whether it is binding in the hydrophilic site or hydrophobic site. And also we are we will be checking whether the ligand is binding exactly to the binding site or not. If it is not talking, then we check with other set of library of compounds, isn't it? So of course the entire structure-based drug design, okay, uses the machine learning algorithms. Here, however, I mentioned for a docking, we have seen so many algorithms in the previous slide, isn't it? So these algorithms are generally used, especially American genetic algorithm for autodock. And similarly, even if you have a gene expression data from the microarrays, and from that, if you want to go for the drug design, identifying a lead compound, of course, here also you need a kind of machine learning algorithms. Okay, to check even how many red color, how many light red color is there, how many green color, how many light green, how many black spots are there. So you and how many yellow color is there, how many dark yellow color is there. Okay, based on these colors only, we can able to diagnose the stage of a cancer. So if and if you diagnose the stage of a cancer, for the stage of a cancer, the doctor can prescribe a drug accordingly. If they are facing the third or fourth, then they have to take a heavier dose. If you see the breast cancer prediction, okay, using the machine learning classifiers, generally, if you see here, they are using support vector machines, which uses linear and polynomial kernel. So if you have positive samples and negative samples, that is positive, I mean, if the patients are infected with breast cancer, okay, so these are shown here. And then if the from the normal patients, so those are not having any breast cancer symptoms, this will be here. So a hyperplane is going to separate between these two different samples. Okay, so this can be analyzed the SVM. And similarly, the logistic regression parameters can be determined from the cancer patients and from the non-cancer patients. And there is also an algorithm called K nearest neighbor. From the K nearest neighbor, we can able to predict how many patients are suffering from cancer, how many not, and even they can predict on the stages of cancer. An ensemble classifier. So if you see, most of the machine learning algorithms are used for a diagnosis and prognosis of cancer, especially here they have used it for breast cancer. So different parameters are there. I told you, even if you want to identify the patient biomarker, like what is the major uh, gene responsible for causing the breast cancer, then they just see from the already existing data sets. I told you nearly three major genes are responsible. Then they will be checking whether each and every gene uh, is uh, is responsible for a particular patient or a combination of genes. All these things they will be checking. Okay. Computational methods, so whatever computational methods in drug design I showed for SARS-CoV-2, the same thing can be applicable for cancer. And for all these drug design methods, we are using a specific machine learning algorithms. Drug repurposing. So if a drug have to be used, like for example, I told you, for we also faced a pandemic situation. Okay, in March 2009, it was kind of the epidemic situation was translated was brought to an epidemic situation. Okay, so at that time, no one know what kind of a drug can be used for treatment of SARS-CoV-2 infection, is COVID-19 for treatment of COVID-19 disease. So at the time, what they did, so they used already existing antiviral drugs. They used the anti-dengue. They have used the dengue drug. They have used the HIV drug. They used almost whatever viruses they know. Okay, but these drugs are not effective, isn't it? Finally, the vaccine came into effect after a few months. Okay. Now, if you see this kind of at that time, initially, not only for SARS-CoV-2, even for the Nipah virus, Ebola viral infections, and other kind of infectious virus, most of the doctors at that time, if they want to diagnose properly, they go for drug repurposing. Even if they diagnose, if the drug is not readily available in the market, so they go for drug repurposing. So if you want to go for drug repurposing kind of software. So even for these tools, you can use the machine learning approaches. Okay, so this is used for both treating the disease as well as for us bringing a therapy. Okay, so different methods are shown here actually for treatment as well as for the therapy. And if you see the artificial intelligence and machine learning approaches, okay, this is most important needed for repurposing drug for different coronaviruses. So for SARS-CoV-2, I told you nearly 100 strains are there. But for the first strain, may COVID-19 and co-vaccine are effective. But how about the other strains? That's most challenging just because of mutations happen in spike glycoprotein, isn't it? Then you have to use the so many machine learning algorithms to find which is the best kind of a vaccine candidate for the specific type of virus. The same apply even for the cancers. So you know the cancers are also hereditary transfer from one generation to the next generation. So and also the cancers are caused because of the mutations. If the cancers are caused by mutations, obviously even whatever drug already prescribed for treatment of cancer can't be used for the mutation. So then you have to again use the machine learning algorithms, and then you can screen the best drug for the particular protein cancer antigen. Okay, can. So this is how we can use this kind of an A and ML approaches. What are the methods here? They mentioned as a machine learning methods. Here they mentioned like support vector machine, random forest, artificial neural network, K nearest neighbor, and deep neural networks. So these are the machine learning algorithms they have used for development of drugs, especially repurposed drugs. Next, they are mentioning about support vector machine. Where can we apply this support vector machine? The support vector machine we also see in this Swiss ADM tool. You remember Swiss ADME tool? This is used for predicting ADME, pharmacokinetic parameters. Before going to this slide, I like to ask you a question. Can someone expand what is ADME? Or I can give you one more thing. Expand LADMET. So in the Swiss ADME, okay, we are using support vector machine as a machine learning algorithm to identify whether your drug is a lipophilic drug or hydrophilic drug. This is, of course, important to check. Okay, if it is more lipophilic, okay, if you are treating a cancer, and for treating cancer, more hydrophilic is needed. If your drug is lipophilic, okay, during the design, okay, or whatever drug you're going to give for treatment of cancer can also target central nervous system, especially brain. Go and target the brain. If you target the brain, obviously they will be getting some numerous kind of disorders. Very good. I got the answer from Veera Rahul, Syed, and Jainab. So the correct answer is liberation, absorption, distribution, metabolism, excretion, and toxicity. So Veera Rahul has completed all the terms. Okay, LADMET. So this is what we call it as a pharmacokinetics, isn't it? Good. So let me take you to the slide. Okay, so if you see a drug here, generally if you see the Swiss ADME, it has to upload one 3D structure of a ligand, or even inputting a SMILES format of a ligand. Okay, you can able to predict not only whether a ligand is a lipophilic or hydrophilic, but also it can predict the drug likeness information and the ADME information. Okay, here they have shown one such figure. We call it as a boiled egg. Okay, if and here in this left graph, you can see there are so many kind of dots in the hyperplanes. There are two hyperplanes. There are so many kind of dots. Okay, so actually, if you see in the top, you find majorly red color, and if you see in the bottom, majorly you can find blue color. So actually, the blue color stands for one particular type of protein, and this is for one type of. For example, if this is a more hydrophilic, the bottom one, the blue color, whatever shown is more hydrophilic, whatever shown at the top is the more hydrophobic. And similarly, even if you see the same kind of results in the boiled egg diagram, okay, you know, in the egg, generally the yolk is of high fat content. So this is what the yellow color, I can tell it is a more lipophilic. And the white color, where you can find the transparent liquid in the broken egg, okay, so where you can find more albumin. Okay, so that is actually more hydrophilic. When compared to yellow color yolk sac, okay, the albumin is hydrophilic. And if your drug is lying in this white, then you can tell it is a hydrophilic nature, whatever drug you are going to use. Okay, so this kind of drug prediction, okay, based on the hydrophilicity parameters can be done using the support vector machine and deep learning models. So where can we apply the deep learning model? The deep learning models, we generally use even neural networks. Okay, in deep learning, we can use artificial neural networks. Then we can call it as a deep neural networks. Okay, here you can find in neural networks how you have generally are going to have an input layer, and you are going to have some layers, hidden layers, like how you find in hidden Markov model. Also, you find one activation function. A neuron is generally is going to have an activation function, going to have some inputs in the activation function, are going to get some output from the activation function, isn't it? But you can also find the hidden layers in the neural networks. So the deep learning generally used for analyzing medical images. For example, if you have a histogram a picture of a liver cancer, you know, in a liver, you have nearly 5 types of cells, cholangiocytes, and some kind of types of liver cells. I can show you. If you see the cells, okay, liver cells, we have hepatocytes, hepatic cells. So see, each and every cell is something looks different, isn't it? Green color, this kind. So each and every, I can show you one. Okay. So if you're finding the histo uh, chemical analysis, you can find these cells or in. So this is actually just a graphical kind of a picture. But how we find the images? You know, if you see the histo, yeah, the images looks like this. Even based on the fluorescent markers, you can find different colors. So you have hepatocytes in one color, Kupffer cells in another color, hepatic status is in another color, sinusoidal endothelial colors in one color. Okay, so all these things, how you distinguish with respect to colors and even the shape of the cells, each and every shell shape is different. Okay, one if you get in the form of a color, this is hidden layer one, and another you can if you can able to distinguish based on the shape, okay, and size, then it is a hidden layer two. Okay, likewise we can use the different parameters, okay, to identify, okay, only from uh, these layers only, and through the activation function, we'll be getting an output. Like if you want to know, if you have a picture. If you have a histogram of liver cell, then you can also able to tell whether is it in what stage of cancer or whether you have any kind of lipid accumulation initially, something. So related to the cells information, what stage of cancer, all these things can be diagnosed using the deep learning. Okay. So just by taking a biopsy of a liver, one can able to tell, predict the stage of a cancer. How it can be done using this deep learning models. K nearest neighbors. In K nearest neighbors generally used for screening the ligands. You remember this? I think on the third session I was explaining you about Zinc 15 database. In Zinc 15 database, we have more than 1.3 billion compounds. If you want to check which is the best compound for a particular target receptor, so you have to talk all these billion compounds with one protein, isn't it? So, but before taking all these compounds for talking, if you have some desired parameters, for example, if you want to select a ligand with a molecular weight 375 and log partition coefficient 2.5. So 375, 2.5. If you see here, you have 20.28 lakh structures. Now you select this 20.28 alone and you download the entire data set. Instead of docking 1 billion compounds, based on these two properties, okay, in X and Y directions, you got some kind of a data set. You can download the data set, and so whatever going to dock, okay, it takes very less time for prediction. Okay, so for this, you can use the K nearest neighbors. Okay, the Zinc 15 uses the K nearest neighbors. And now we can go for random decision forest. Where can we use this random decision forest? Especially for QS, quantitative structure activity relationships, we are using this random decision forest, which is a form of a decision trees where multiple samples of data are processed by decision trees and the results are aggregated in the form of training data set. So in QSR, you're going to have a training data set, and for the unknown compound, the activity is predicted. So that's what we call it as a test data set. Machine learning techniques. I told you the machine learning techniques like artificial neural networks can also be used for uh, prognosis or diagnosis of a cancer. Okay, for example, if a person is infected from lung cancer and if you want to identify, this is an example for a COVID-19, of course, COVID-19 infections in the lung, but even the same is applicable for the lung cancer. Okay, if a person is infected from lung cancer, obviously they can find abnormal tissue proliferation in the lungs, isn't it? So whatever X-ray images they will be getting, okay, they just pass it to the software to get the heat maps. From the heat maps, wherever you get this yellow color, red and yellow color, here, I think if you see from the first column, you see the third row, you find more yellow. When compared to the third row, fourth row line, no yellow is maximum. So it means that they are in final stage of cancer. So if they are in the they have more infection, cancer infection, you find it in yellow color. But if it is in the black color, something like that, or dark red, something like that, then they have less cancer. It can be a second or third stage, or it can be even first stage of cancer. So based on the colors in heat maps, you can predict. Not just this gray and white images can give you an idea, but using the heat map, you can get somewhat more precise information. For this, these softwares are used. And machine learning, in overall, I can tell you, it is a subset of an artificial intelligence. So many machine learning algorithm methods are there used for different drug design studies. And even one such kind of a database developed, and even we can develop a few more databases using R programming. R programming is meant for statistical analysis, especially. That's what you understood, isn't it? But in an article, the R programming is also used for drug candidate selection. If you have some thousands of compounds, you want to select which is the best compound for a target receptor, still we can able to develop your own tool through R programming. So there are plenty of libraries. For example, in C programming language, you have output function like printf and input function like scanf present in stdio.h, isn't it? Standard input and output header file. Similarly, there are also numerous functions present in this library packages. Like how you find hash include stdio, similarly you find libraries. Libraries for RCTK, chemometrics, if you want to work on chem informatics, if you want to use a Java to get the 3D structures in the visualizer, ChemMine R, if you want to mine the data from a website, and if you want to get in the software automatically, you have to use this library cluster. If you want to get in the numerous compounds, you have to cluster it, RGL, vegan, facto, extra, fingerprint. So there are numerous kind of libraries. If you can include all these libraries, and you can use the functions present in the libraries. So the R programming, they have developed so many libraries to work on selecting the drug candidate. That is, if you want to develop a virtual screening, high throughput screening, or lead hopping tools, we can able to make it out using the R programming. This is the paper published in 2020. MATLAB, this is also a well-known machine learning tool, isn't it? So, of course, the students and faculties of SVC are having an individual license. Okay, so here, fortunately, here we have a free license applied to all the students and faculties. So we can also, if you want to practice on machine learning, this is one of the best to work on. So whatever is mentioned, okay, about the types of methods, all these things, even you can find in this MATLAB software. This is an extract I have taken from the MATLAB software directly. And there is one such module called TensorFlow. Okay, if you see, MATLAB is generally linked with the deep learning toolbox, and similarly, the Python is linked with the TensorFlow. The MATLAB is also linked with the TensorFlow. Not only the Python is having the TensorFlow, MATLAB is having deep learning toolbox also. That is, you can also link with the cloud computing. Okay, that's what I mean. MATLAB is almost linked with almost all the programming languages and matched with a few softwares for interactions. If you see this MATLAB online courses, if you have a MATLAB license, so one can go to the MATLAB software and you can search for the courses. We have machine learning and deep learning courses. So just 6 modules, 2 hours. So very easy to complete. If you complete this, obviously you can also get a free certificate. Okay, like however you can find Coursera, there are some courses present in the MATLAB software itself. Okay, so you have MATLAB both online and offline. Okay, if you don't have sufficient data in your laptop or in a system, instead of downloading the entire MATLAB in offline, you can also use the MATLAB in online with your account. And MATLAB using MATLAB online course. So what are the major modules present? So in machine learning, if you see, you find nearly 6 modules. Okay, so these 6 modules are easy to complete. So anyone can go to this and can complete to get a certificate. And if you want to know more about how to practice machine learning, okay, so much detailed information is available in W3 schools, like the starters, those who are scared on working on programming. But if you want to learn something new, which is helpful in future, obviously you can go to this kind of a website, W3 Schools. Okay, so these are some of the references. Now I can take you to the MATLAB software. Okay, so in MATLAB, to open the MATLAB, you simply have to type MATLAB. I just installed the MATLAB 2024. Okay, so if you open the MATLAB 2024, you get a window like this. Okay, if you want to see, this is one such report. I think I can open from the beginning, then you can understand. MATLAB. So this is the 2024 version, 2024A, recently installed. I think it's opening. MATLAB. Yes, it's getting open. Actually, for installing this software, it may also consume 25 GB. Okay, based on what models you want. So before installation itself, it asks the user preferences. So you have to select what tools you want. We are working on biotechnology principles, isn't it? So there is one toolbox called bioinformatics toolbox, and if you want to work on machine learning, even you can install the corresponding machine learning toolboxes. Okay, so this is how a MATLAB tool looks like for the freshers. If they want to learn something about MATLAB, okay, so you can learn MATLAB by clicking here. Okay, if you click Learn MATLAB, it directly takes you to the online website. Of course, you should need an internet. So, however, I mentioned to learn the online courses, and if you want to work on a few modules, so all these things you can able to find from here. Okay, Learn MATLAB, request support, community, like help however you can. If you don't know about anything, like if you want to know about MATLAB code, MATLAB examples, okay, all these things you can learn from here. So, for example, I'm going to click examples. So you will be getting numerous examples. So exactly what you want to work, you can select accordingly. Okay. So we can find in the left. Okay. So we can select accordingly. Okay. This is something about how to use the MATLAB. Okay. Now, if I want to work on specific module, okay. So I'm checking here plots, apps. In apps, we have one kind of tool called Biology Model Builder. Okay, so I'm going to click this Biology. So this is actually systems biology approach. Okay, I told you if you want to develop a drug design in cancer, obviously you have to use system biology approaches, isn't it? So to work on system biology, this is one of the best software tool. What is that? The MATLAB. So now I'm going to open an already existing model. So for this, I click, if you want to create a new model, click a model. So you have to develop a training data set. Okay. But if you want to open an already existing model, that is an example, click open here. So if I click open here, it's asking to locate a file. So just because I've installed MATLAB, I think it's present in the program files. I go to the program files and check for the MATLAB. Uh, I think I have some. Okay, MATLAB. I have versions. Earlier I installed 2020, now upgraded to 22, and 4 days before I upgraded to 24. Now 22, I have some examples downloaded in system biology. I'm going to the example system biology folder. Inside the data, I have one model called tumor growth model. Okay, so if you are working on a tumor in animal models, in rat or mice, okay, so these models are already developed as an example. So just like that, you have to open the model and whatever properties you are getting in your in vivo experiments in lab, just feed the properties, feed the values, feed the values. Okay, so see here. So this is how a MATLAB tool looks like. Okay, in the MATLAB tool, For example, if you take my kind of a research, I am recently working on developing a drug for type 2 diabetes, that is T2DM. So I'm going to use a single drug, which not only going to target type 2 diabetic mellitus, but also linked with NASH, non-alcoholic steatohepatitis, or linked with NAFLD. So NAFLD is, of course, a second stage of liver cancer. If you want to use a drug which can target the type 2 diabetic mellitus as well as a kind of a second stage, okay, so then I can also use this kind of a pharmacokinetic model, PK model. So here, if you see if my T2DM is the major disease, okay, then in the central component, you can use a pancreas because the T2DM is major dependent on insulin secretion. Of course, the major organ, if you take, it's a pancreas, isn't it? And if you see the peripheral component, okay, that is a liver. And so if you want to check what is the effect of a drug, that is a drug used for treatment of T2DM in association with a liver kind of a disease, then obviously a peripheral component is your liver. And we can also able to develop a tumor growth models. If this is a normal kind of compartment, also we can develop for a tumor growth models. So in a sample, they've already provided this. Okay, if you want to change the information, you can change the information here. So what is the value? How much drug you like to you're going to? So this is like a simulation. So if a human body is taking a drug, okay, 3 doses a day, so you can also feed accordingly. So what is the dosage per session? And if 3 doses are there, you have to feed 3 doses. Okay, so all these informations are available here. Okay, here you have all the thing. Okay, and even the kinetics. If you know the kinetics, even you can fix the kinetics here. Okay, so the K1, K2, so all this the first order, the forward reaction and the reverse reaction rate constant, and what is the value with respect to the units. All these things are mentioned. If you change the values, obviously you can find a kind of a graphical change, graphical alterations. So now whatever values already we have in the sample, if you want to see a kind of a graph for the developed model, simply click model analyzer. There is one option called model analyzer. Now I click it. We got like this. Here in the model analyzer, okay, we got what is the drug they are using and how many days they have to prescribe. Okay, and what is the simulation time? So what is going to happen with the if this drug, this concentration taken for the this much doses and this much days, then obviously get a kind of a response. If you click there run, there is a button called run. So whatever parameters you already fed in the SimBiology tool, okay, you are going to get a graphical output. So something like what is the bioavailability of a drug with respect to time. So that is going to show here in the model. Okay, if you run this model. So this is the graph actually. Okay, for how many days and what is the drug response. So all these things we can able to get. They have given passively tax cell for this they got this kind of a response. So how about the central component and how about the peripheral component, the drug accumulation? So and how about the normal W? So this is something about um, working on PK models, which majorly uses each and every step you're using the machine learning algorithms. Okay. So I have completed my session and also I've taken you to the MATLAB. And when you have any doubts, you can ask me. So now I think I can stop recording. Thank you very much, participants.